My project is on the depth preferences of Cambarus genus crawfish. North Carolina's streams and brooks are host to many crawfish species, and among these, the genus Cambarus contains the most vital members to the overall ecosystem of said streams. Some of these, like the Appalachian brook crayfish and the big water crayfish, are the largest crayfish in their habitats, with their advanced size and potential age making them superb indicators of the long-term quality of the stream. Therefore, measuring their preferred level of depth echelon, or how deep they choose to stay, can lead to better understandings of the qualifications of a healthy, supportive stream. This study aimed to find such data. 5 streams of comparable depth were sampled daily for 2 weeks in order to determine the size and depth of Cambarus specimens. This data was then analyzed for trends. The resulting data purveyed a strong positive correlation between size and depth, with the older, larger individuals preferring to stay at deeper echelons than the smaller, younger crayfish. This is cause for inference upon the importance of the presence of deeper pools in the streams being either built or restored, as it can be surmised that Cambarus crayfish required the depth to survive to considerable sizes and ages. The focus of this study is crayfish. They are a freshwater clade of crustaceans anatomically similar to lobsters, their marine sister group. In particular, the aims of this study focus around those of the genus Cambarus, a large genus composed of many of the largest and most impactful members of their ecosystems. 2 specific species were utilized in the study due to their relative abundance and ecological role: Cambarus bartoni and Cambarus robustus. Respectively, that is the Appalachian brook crayfish and the big water crayfish, respectively. These were chosen for their massive ceiling for growth and age, with both approaching closer to 100 millimeter in their late adulthood compared to other Cambarus species which max out around 50 to 70 millimeters, and their status as an umbrella species. For the sake of this study, umbrella species were defined as those whose conservation enables the conservation of others with greater ease. Their role as an umbrella species extends into their role as both a keystone species and a bioindicator species. As a keystone species, both Cambarus bartoni and Cambarus robustus are of immense importance due to the role they play in the transfer of nutrients and energy from one ecosystem to another. They are the final stepping stone in the process of nutrient and energy cycling from the freshwater stream ecosystem to the surrounding forest ecosystem, and represent the top of their food chain and the bottom of the forests. As bioindicators, they serve 2 major functions. First, as was reported in the 1991 paper by DiStefano et al., they are major indicators of pH imbalances, especially in terms of increased acidity, and due to their high resistance to mortality events in such cases of acidity, they display numerous subfatal signs of pH damage and illness, which allows them to be a sign of risk for further acidification, and that allows that to be assessed and prevented before the loss of the crayfish species occur. Second, as was put forth by an experiment designed and conducted by Sherba et al. In 2000, they are equally excellent indicators of heavy metal poisoning, such as that is copper, which can leach into waterways by means of pipes or by means of wiring. For the sake of this understanding, a few terms need to be clarified before I move into the methods. While discussing the flow of streams, riffles and pools are often mentioned. Riffles are shallow, typically rock or sand-filled regions which cause oxygenation and filtration to occur as the stream passes over them. Following immediately after riffles are pools, which are deeper sections caused by the erosion of the heightened water speed. These often are found at bends in a stream or at the base of waterfalls, cascades, or just tall riffles. Pools themselves contain varying depth levels, in this case organized into depth echelons, which are layers at which crayfish aggregate and live, with distinct levels of separation. Finally, the hypothesis of this study is that there will be a positive relationship or correlation between the size of the observed specimens and the depth they occur at. The methods of this study were relatively simple, and to collect and analyze the correlations between Cambarus genus crayfish size and the preferred depth echelon, a 2-fold system of data collection was employed, though it was truly really 3 stages, but for the sake of simplicity, I'll break it down into 2. I collected data and then organized the data to analyze it for trends. And then I considered some confounding variables, though there were very few to actually consider, considering the closed-off sections of streams I picked. The first of the two main data collection procedures was the measurement of the depth at which the specimen resided. 5 stream locations were selected based upon the presence of a rock riffle pool system and the presence of a stable camberous population of either Bartoni or Robustus species. Each location was assessed for depth echelons or step-like layers of rock which provide space for specimens to alight, and then accordingly mapped on a topographical depth chart. The specimens in each of these locales were then recorded daily for 2 weeks, with their depth being catalogued in centimeters below the surface. Collections took place during the peak of crayfish activity, typically falling within an hour of dawn or dusk, and were minimally invasive. As the depth of the locales were already calculated, each crayfish were undisturbed during the collection of depth data, as an accurate approximation of position could be made and then related to the previously constructed depth charts. This ensured that the presence of perceived predatory threats didn't cause them to aggress to lower echelons by any of the specimens, which lends greater credibility to the results gleaned. The second measurement procedure was the collection of length data. This was done in a similarly minimally invasive manner, with the width, length, and perimeters of each depth echelon being recorded prior to conducting collection to allow for non-invasive measurements. Utilizing pixel scaling from a surface with a known length, digital photographs of each specimen were approximated for length with an accuracy of within 1 millimeter. Flat-angled, top-down photographs were utilized in all possible opportunities, and minimal angled photographs were taken to reduce the chances of mismeasurement due to lens warping. Each cataloged length corresponded to a cataloged depth of a specimen, allowing trends to be assessed. Depth charts of each location were constructed to demonstrate the general layout of echelons. A scatterplot was constructed to demonstrate the relation between the variables of depth and size. From these tables and the data collected, the results and conclusions therein emerged. This is just a simple representation of the depth charts of all 5 stream locations. This is figures A through E. As you can see in the figure key, not all of these shared both species. Some of them only had one, such as figure A and figure C, which were locations 1 and 3 respectively. Location 1 only had a population of Camberus bartoni, though it was a relatively sizable location, so still deemed fit for being used. And stream location 3 had an equally large population of Camberus robustus, or the big water crayfish, just for reminders. All the rest of them had both. In most of them, it was a rather equal distribution. And the 4th stream location, there seemed to be some sort of an imbalance. There was a lot more robustus than there was bartoni, but that wasn't present in any of the other streams. And this is the trend of the overall data, and as can be seen in this regression line, the results do show a conclusively correlated relationship between the length of a specimen and its chosen depth echelon, with a positive relationship showing that those of a greater length prefer deeper sections of the stream pools they inhabit. This is true across the entire range of lengths, with those closer in size to the smallest recorded specimen of 25 millimeters, preferring shallower sections than those closer to the largest recorded specimen of 98 millimeters, and those preferred deeper sections. Figures A through E, the maps on the previous slide, showed the depth layouts of each selected stream locale, with echelons being conveyed as 15 centimeter deep layers. In the case of most of the streams that I chose, this was actually rather accurate to the geologic format of the stream itself. Most of them were arranged in close to 15 centimeter rock shelves as they reached toward the bottom. There were some exceptions, but for the most part, those exceptions were nullified by the fact that the crayfish stayed on the shelves rather than the smoother sections that didn't have clear distinctions. Among all the stream locales, the average preferred depth echelon lay at about 36.2 centimeters. This is in line with the average length of 72.7 millimeters, as both lay at a major clustering of medium-sized, medium-depth specimens as seen on the graph. The positive correlation depicted holds an R2 value of 0.81, making it an accurate representation of the correlation. Thus, in combination with the strongly positive slope, it can be assumed that this trend would continue over larger sample sizes. Due to the relatively large sample size of 50, contributed by a multitude of locations, conclusions can be extrapolated from the data found onto a larger population to some degree. The analysis of the conclusions drawn from the data is as follows. So while this is at most a correlational finding and cannot conclusively lead to any indications of causation, inferences and predictions can nonetheless be made. One such inference is that due to the heightened preference for deeper echelons among larger individuals, deeper pools are a requirement to sustain older, more sizable populations. It may also be inferred that these individuals are able to grow to such sizes and ages due to the available depth. The darker, harder to reach echelons towards the bottom of the stream may present a sort of visual impairment to surface hunters such as raccoons and herons, which are the leading surface predators of Cambrous genus crayfish in small streams that lack the fish large enough to tackle them, allowing lessened predation pressures and greater success at survival at a great age. This could be bolstered by experience, as those who discover this benefit would be more likely to organize on the lower echelons in the future, which would allow them to survive to greater extents than those who would remain close to the surface because they would lack the experience. That would lead them to conclude that they need to go deeper to escape predation. This too could also partially explain the trend seen in the collective data. And these conclusions could be applied to modern attempts at watershed engineering, particularly in the field of reconstructing streams or fully creating a whole new stream. Utilizing riffle pool theory, deeper pools could be designed with faster flowing riffles immediately before so that they can maintain their depth over time and not be filled in with sediment, and to provide adequate habitats for more successful crayfish populations. This can actually be seen on campus in places such as Rocky Branch, where features such as deep pools and crevices have been formed either with rocks or with borders keeping sediment out. This allows for a larger population of larger individuals, and thus it allows the roles of the Cambrous genus to be fully realized to a fuller extent. With a healthier, stronger crayfish population comes the benefit of a stronger umbrella species population, allowing the betterment of conditions for all organisms sharing that environment. Alongside this, such a population would allow the bioindication of potential pollutants to properly be assessed with greater ease and speed. Agar on the right is one of them I found. That is an Appalachian brook crayfish, which is the Bartoni species. It was pretty cool. These are my citations.